Okay, do we see the candidate? Mr. Zhang. You are present. I cannot see. Now I can see you. Welcome. Welcome, Mr. Zhang.
Thanks.
To this ceremony. Also welcome to your family and friends who might join the streaming session. And I now hereby open this academic ceremony in which you, Mr. Zhang, will defend the academic thesis entitled DMT1, a novel regulator of physiological notch signaling and radiation sensitivity. Before I invite you to present your major findings, I would like to introduce your supervisors, Professor Mark Voice. He's a professor of tumor, let's say his expertise is in tumor microenvironment and therapy response, working at the Department of Radiotherapy, and Dr. Keulers. He's an assistant professor at the Department of Radiotherapy. Now, Mr. Zhang, can I invite you to present in a short presentation of about 15 minutes your major findings and major conclusions? Please, go
Yes,
ahead.
of course. I will share my PowerPoint. Okay. Dear proctor, dear opponent, dear supervisors, colleagues, and friends, welcome to join my online defense. In the next 15 minutes, I would like to defend my PhD thesis. The title of my PhD thesis is DMT1, a novel regulator of physiological notch signaling and radiation sensitivity. Firstly, I would like to introduce my research background. Well, our human bodies are composed of loads of cells. Between them, there is crosstalk between cells. Well, for notch signaling, it is one of the most effective ways for cells to share information with each other. The dysfunction of notch could cause severe disease like cancer. Well, about cancer cell, their cell, the growth of them are not under control, which could damage the growth of normal cell and even the health of our human body. The understanding of notch, especially the modulation of notch, would contribute to the treatment towards cancer, especially notch-dependent cancer. The crosstalk through notch signaling could achieve intracellular cleavage. The production notch protein can reach nucleus, and then notch protein can interact with DNA binding protein. This interaction could activate the notch target genes. During this process, there are some intracellular vesicles which could take part in the notch protein delivery. For lysosome, it is one of the intracellular vesicle. Notch protein can be digested by lysosome. In addition, notch protein can also reach lysosome. For lysosome, it is organelle which have some specific characteristics. Firstly, the pH of lysosome tend to be low. In addition, there are some enzymes in the lysosome. The enzyme could degrade the trash from the cell. Previously, my colleagues found that the deletion of DMT1 could inhibit notch. For DMT1, it is iron transporter. While for cell growth, the cell needed to uptake iron from the environment. The uptake of iron from the environment could contribute to cell divide, growth, and the formation of cell population. As you know, the lack of iron could cause severe disease, like anemia. DMT1 just plays an important role for cell to absorb iron from the environment. In addition, DMT1 can be expressed in different places of the cell. On the plasma membrane, DMT1 can be expressed there and help cell to absorb iron. In addition, DMT1 can be expressed on the membrane of lysosome. Well, the expression of DMT1 there could help the lysosome pump out the iron. Well, for me, my research question is how DMT1 could modulate Notch signaling. Importantly, I should try to explain why without DMT1, Notch was inhibited. In order to answer those questions, I read some papers. I summarize what I found into chapter 2. It is a review. It's relevant with lysosome stability, degradation, and repair. In addition, I summarize the modulation of DMT1 on Notch signaling, and I explain the reason why without DMT1 Notch was inhibited. What we found was summarized to chapter 3. During this study, we're curious about whether DMT1 deletion could influence the radiosensitivity. We found the phenomenon. We explained the reason behind. What we found was summarized into chapter 4. Next, I will mainly introduce the work in chapter 3 and chapter 4. For the work in chapter 3, firstly, we use a dye to measure iron level. Well, from the result you can get without DMT1, free iron in the cell was upregulated. High iron in the cell, which can take part in the reaction and then generate ROS. With the dye, we found that without DMT1, the ROS level was upregulated. About ROS, we can regard it as a toxic material, which can damage the component of cell, including lipid, protein, and even the membrane of the cell. Well, in order to further study the ROS damage, we use a dye to measure it. This is the image from conferral microscope. The green signal here means the damage level. The increased intensity means the damage was enhanced. From the image you can get without DMT1, the ROS damage was induced. Well, in order to further study what ROS could damage, well, lysosome plays an important role in the Notch signaling. So we started to analyze the damage on lysosome. Firstly, we use a dye to measure the pH of lysosome. Well, in the normal cell, this dye is sensitive toward pH. The low pH there, we can get the signal. From the red, we can get that, well, the pH is low. But in DMT1 knockout cell line, it's still the red signal just be decreased, which means the pH of lysosome was changed without DMT1. Well, the change of lysosome pH might influence the enzyme of lysosome. The lysosomal intracellular activity depends on low pH. In order to measure the lysosomal intracellular activity, we use the substrate, which can be digested by lysosome. From the result we can get without DMT1, well, lysosomal intracellular activity was downregulated. Well, in order to understand whether lysosome damage could influence Notch, we use a chemical which can target lysosome and damage lysosome. From the result we can get, the chemical treatment can inhibit Notch, which means, well, lysosomal damage can cause Notch inhibition. Well, importantly, to further understand why without DMT1 Notch was inhibited, we started to analyze the Notch protein distribution in the cell. This is the image from conferral microscope. The blue signal means nucleus. The green signal is from Notch protein. From the image, you can find the blue signal can be overlapped with green signal, which means without DMT1, Notch protein can still reach nucleus. But importantly, when we analyze the interaction of Notch protein with DNA binding protein, we found that that interaction just be inhibited. This result just explains why without DMT1 Notch was inhibited, because this complex formation was inhibited. In this way, Notch target gene cannot be activated. To summarize my whole research story in chapter 3, without DMT1, free RNA in the cell was upregulated. RNA in the cell can take part in the reaction and then generate ROS. Without DMT1, lysosomes was impaired. As a result, Notch cannot interact with the DNA binding protein. This just explains why without DMT1, Notch target genes can be inhibited. Well, during this study, we're curious about whether DMT1 deletion could influence radiosensitivity. About radiosensitivity, it's the sensitivity of the cell towards radiation. After radiation, if the cell dies easily, we tend to think the cell has increased radiosensitivity. Well, then we started to study the radiosensitivity of DMT1 wild type and knockout cell line towards radiation. From the result you can get, after radiation, DMT1 knockout cell line tend to die easily, which means without DMT1, the radiosensitivity was increased. In order to understand the reason behind, we use ferroptosis inducer to treat the cell. About the ferroptosis, it is RNA-dependent cell death. There are some characteristics relevant with ferroptosis. Increased RNA and vesicle tend to be ruptured. When we use this chemical to treat the cell, DMT1 knockout cell line tend to die easily. Well, interestingly, when we use ferroptosis inhibitor to block ferroptosis process, we found that ferroptosis inhibitor treatment could rescue the surviving of DMT1 knockout cell line. Well, to summarize the figures here, we find a new cell state. Well, for normal cell, the cell tend to stay far away from ferroptosis, from cell death. But for DMT1 knockout cell line, the cell tend to stay close to ferroptosis. There are some characteristics relevant with ferroptosis. Increase RNA, increase ROS, but the cell still keep alive. Because of this point, the cell tend to be more sensitive towards radiation. Importantly, we assume that the deletion of DMT1 could increase the radiosensitivity, contributing to cancer radiotherapy. In order to confirm this point, we use DMT1 inhibitor to treat cancer cells and then analyze the radiosensitivity. From the result you can get, DMT1 inhibitor treatment can increase the radiosensitivity of colon cancer. In addition, when we use DMT1 inhibitor to treat breast cancer, well, after radiation, the breast cancer cell line tend to die easily. Well, those figures just confirm our assumption that the inhibition of DMT1 would contribute to cancer radiotherapy. To summarize my whole research story in chapter 4, without DMT1, our cell tend to be more sensitive towards radiation. Well, our cell has some characteristics relevant with ferroptosis. Increase RNA, increase ROS, but our cells still keep alive. We define it as pre-ferroptosis. Well, we believe that the deletion of DMT1 could induce pre-ferroptosis, contributing to cancer radiotherapy. Our findings just provide the clinical potential. With those research stories, I would like to summarize my PhD thesis into one sentence. RNA metabolism and lysosomal health control Notch signaling. Well, and this determines how cells live, die, or respond to radiation. I would like to give the word back to proctor. Well, thanks for your attention.
Thank you, Mr. Zhang, for this clear overview and nicely illustrated presentation. Your thesis, the manuscript of your thesis was already assessed by a number of experts that were selected by your supervisors. Their judgment was positive, and that's why you're now able to defend your thesis, because there are always questions that remain. And we would like to see now in this opposition and defense whether you can defend your work and explain your work in an academic fashion. And that is why now the opposition will start with Professor De Wind. He is a professor of molecular cardiovascular biology at the Department of Cardiology at the Maastricht University. Professor De Wind, floor is yours.
Thank you, Pro-Rector. Dear candidate, congratulations on this festive day, yes, where you celebrate a big accomplishment. I really congratulate you with a pleasantly written thesis. Beautiful presentation as well. Yes, was very well done. Of course, I also salute and congratulate your supervising team, Dr. Keulers, Professor Voice, and of course also congratulations to family members and loved ones because you know a PhD graduation is a journey that you don't take all alone. Dear candidate, I was really intrigued with the cover of your thesis, and it brought me back to my own youth when I was still studying French in high school, because it really looked like the illustrations of a French novel that's called The Little Prince by Antoine de Saint-Exupéry. That deals on a pilot that crashes in the Sahara Desert and encounters a little prince from an asteroid, and he recounts the stories of his travels to different planets. Is perhaps was your inspiration this French novel or not at all?
Actually, from this cover, you can find that it seems that I fly from a long distance and then I reach the cell. Actually, during my study, I can really find that the cell seems like, well, a planet. There are a lot of information there. Yeah. Well, gradually, I think even the cell is small, but the information seems like the universe. Yes. So,
yeah. Okay. No, that's beautiful. Okay. So, but I really, after your defense, it might be something to pick up that book. It's a very famous French novel. But anyway, I was not hired to ask questions about French literature or you know, tiny princes. But let's immediately dive to page 71 in your thesis, chapter 3, and you show there that in Figure 1D. In your DMT knockout 1 knockout cells, you show that basal ROS levels are increased. Reactive oxygen species are increased compared to the wild type cells. Yes?
Yes, yes.
And but then you treat the cells with peroxide, yes, H2O2, and the amount of ROS does not further increase. So in the DMT1 cells, they're higher than wild type, and you cannot increase it further with hydrogen peroxide. And I was thinking at least half an hour about this experiment and the implications it might have. How do you interpret this finding? What is going on there? Is this a technical issue or is this biology?
From my understanding, you mentioned that hydrogen peroxide cannot increase the ROS level in diameter knockout cell line furthermore, right? Yes. You want me to explain the reason behind?
Yes.
I think, you know, I tend to believe the ROS level there might reach the threshold for diameter knockout cell line. The without DMT1, well, the antioxidant capacity might be impaired, and the cell cannot clear out the hydrogen peroxide. But well, if the ROS level was increased furthermore, I think the cell tend to die easily.
Okay.
Finally, we can still measure it, which means we only keep the alive cell there.
Yeah. So you don't think that this is a technical issue that has something to do with the probe, the fluorescence of the probe that can no longer, that is not linear enough, you're not in a linear range, or you're saturating the system in a technical fashion? You really think this is the biology speaking?
Yes.
Okay, good. So then let me go to my final question, because time really flies, and that is, with a DMT1 inhibitor, you showed quite convincing to me that cells become more sensitive, cancer cells become more sensitive to radiotherapy. Yes?
Yes.
And now let's zoom out. Imagine you have a cancer patient.
Yes.
How do you want to translate this finding? How would you like to create a DMT1 knockout cancer cell to treat patients so that they respond better to radiotherapy?
If we really want to transfer what we found into clinical study, especially for
that's the question.
The radiotherapy towards cancer patients, I think we can use DMT1 inhibitor to treat the cancer before radiation, just to try to induce ferroptosis before radiotherapy.
Yeah, so wouldn't you worry to just give a patient DMT1 inhibitor, a small molecule, then all the other cells will also become slowly DMT1 knockout?
Yeah, actually, this is a very good question. I don't think every cancer cell is good for the usage of DMT1 inhibitor. We should choose specific cancer cell types based on my own result. Well, DMT1 inhibitor can only increase cancer radiotherapy in some cancer cells. So we need to choose specific cancer type, especially with high iron, high lipid peroxidation there.
Okay. Hey, I would love to continue the discussion with you, but I'm looking at the clock and I'm looking at the pro-rector, and he's also nodding. So I give the word back to the pro-rector. Thank you for your answer.
Thank you. Nice word. Thanks.
Thank you, Professor De Wind. The next opponent is Professor van Kempen. He is Professor of Molecular Diagnostics at the Pathology Department at the Maastricht University Medical Center. Professor van Kempen.
Thank you, Mr. Rector. Dear candidate, I also have read your thesis with my pleasure. I would like to congratulate on the product that you've made, and also include my congratulations for your promoter and co-promoter. It's a challenging topic, especially because you're looking at two factors, the DMT1 and notch signaling, whereas there's a quite complex signaling process going on. And I got a bit confused because in chapter 4, you mentioned that DMT knockout cells, the mouse embryonic fibroblast, are cells in a pre-ferroptosis state, right? So that would argue to me that would signal to me that there's quite a lot of free iron that might induce a ferroptosis response. And on page 79 of your thesis, you state that your research has linked notch receptor regulation to cellular iron metabolism and proteostasis. That sounds to me a bit counterintuitive. And I started to wonder, and I started to read in your chapters and look for the evidence what the role of iron might be here. What is the evidence that either free or endosomal sequestered iron or iron in the iron storages are actually playing a role here? Do you know what iron is actually contributing to the whole effect that DMT is doing too much?
Highly esteemed opponents, thank you very much for your nice work. For iron metabolism contribution, I think without DMT1, the iron in the cell was upregulated. Iron could take part in the faint reaction and then generate ROS. For the ROS, it could damage the LAMP1, the lysosome by the induction of lysosome by the induction of lipid peroxidation. We have the figure to show the ROS could damage lysosome by the induction of lipid peroxidation. But notch protein, the degradation of notch, depends on lysosome. So
can I interrupt? Because iron has 3 subcellular localization. It's free iron within the endosomes that ultimately cycle through a lysosome, and there's iron in the ferritin storages. Right. So if you talk about iron, what iron are you actually discussing here? Is it iron?
It's 2+. 2+.
Iron 2+ either as free in the cytosol, iron 2+ in the endosome, or iron sequestered in the iron storages?
Actually, we use a dye to measure free iron. It's free iron. We didn't measure the iron in specific endosome. We only focus on free iron in the cell.
Yeah, so how do you then exclude, or can you say that the other iron sources do not play a role if you have only measured free iron?
Actually, we have some figures relevant with free iron, 2+ iron, not relevant with 3+ iron, right? What figures are they? Well, ferroptosis is 2+ iron dependent cell death, right? In addition, for ROS and free iron, 2+ iron can take part in fainting reaction and then generate ROS, not 3+ iron. So our figures always relevant with 2+ iron.
So 2+ iron is only cytoplasmic and it's never in the lysosomes?
No, DMT1 isoforms can also be expressed on the endolysosome.
Yes.
So 2+ iron can also reach endolysosome. Yes.
So if you would be able to extend your PhD project, how would you discriminate between the localization of the iron molecules then in the cells where you start to either inhibit DMT1 or start to inhibit gamma secretase or whatsoever? What type of techniques would you use to demonstrate it's the free iron 2+ in the cytoplasm that's actually doing what you think is happening in the cell?
Actually, we can use confocal microscope to check this point. There are some different markers for different vesicles, like early endosome, late endosome, and lysosome. Maybe we can use a dye to target RAB5, this marker for the endosome. If we can also target LAMP1, it's the marker for lysosome. And then we can use a dye to target the iron, right? And then we can do colocalization analysis. In this way, we can measure the distribution of iron in the cytoplasm or in different vesicles.
Thank you. That was the answer I was looking for. Looking at the time, it's time for me to give the word back to Mr. Prorector. Thank you very much, Mr. Zhang.
Thank you. Thank you.
Thank you, Professor Van Kempen. Then the opposition will be continued by Dr. Milot. She is Associate Professor and Principal Investigator at the Experimental Cancer Department of the Department of Pathology at the Maastricht University Medical Center. Dr. Milot.
Okay, thank you, Prorector. Oh, dear candidate, I also want to congratulate you with your thesis. I really enjoyed reading it, but I also have to say it's a challenging topic. I also really want to compliment you with the cover that I really, really liked. I didn't look at the history of the cover, so I will not discuss that with you. But I think it's really nice how you showcased your thesis with this cover. Now, I was mainly interested in the therapeutic potential of DMT1 in cancer. So I want to focus on chapter 4. And I was wondering, like, because your main aim in this chapter is to look at the therapeutic relevance, why you use throughout your chapter mostly the mouse embryonic fibroblast, and only at the end, in figure 6, you started to investigate the inhibition of DMT1 in cancer cells.
Highly esteemed opponent, thank you very much for your nice word and question. Actually, at the very beginning, we are very curious about whether the deletion of DMT1 could increase radiosensitivity. Firstly, we find this phenomenon, and then we try to understand the reason behind. Well, if we can really explain this point, gradually, we think, well, maybe it's a potential, it's a clinical potential. So at the final figure, we started to think, to test at this point, whether the inhibition of DMT1 could contribute to cancer treatment. So
yeah,
this
so maybe
is
it's
logical.
Maybe it's the sake of time for your PhD time that you had, but you only look there at the clonogenic potential, while you also already described throughout your thesis that DMT can really have different functions in different contexts. So I was really wondering, like, why. I was really interested in reading it, like, when you use this inhibitor in the cancer cells, what is, for example, happening in ferroptosis and all the other other outcomes that you measured in the fibroblast? Like, weren't you interested to see what is happening with that in the cancer cells, or, for example, the notch signaling? So you only focus on this clonogenic potential, and I was really eager to learn more.
Yes, actually, if I have time, there are still a lot of things which I would like to do. With the mesh cell line, I did some experiment to measure the ROS level, the lipid peroxidation, the GPS4 expression, the autophagy flux. Actually, in cancer cell line, if we could also repeat the phenomenon for the mesh cell, it's just a strong evidence to it just make our conclusion consistent. So in the cancer cell, the next step, I really want to analyze the lipid peroxidation from different cancer cells. In addition, the free RNA, I also want to measure it, because this can really influence the sensitivity. Some cells might become more sensitive, but some cells might not very sensitive. So I really want to explain the reason behind, but I don't have enough time now.
Okay, yeah, it's a time issue that I understand. So you have interesting, like you already discussed previously, you have interesting data in mainly the colorectal cancer cell lines, where you see an effect on radio sensitivity. So if you would have more time for your next step, would you also think it would be interesting to discuss this or to study this further in a vivo model?
Yes, actually, I choose 3 cancer types: colon cancer, breast cancer, lung cancer. If I have time, I really want to construct colon cancer mouse model, because in my the radiation experiment, I choose 2 cell lines for colon cancer. When I use DMT inhibitor to treat them, both of them, the radiosensitivity can be increased, which just make me feel very confident. If we can construct the colon tumor mouse model, maybe we can also achieve a good treatment effect.
Yeah, and as you probably know, like, if you would go to an in vivo situation, there's also the tumor microenvironment. You have other cells like macrophages or different immune cells, which I feel, because I really like this cell-cell communication in the tumor microenvironment, that's my expertise. But so I was a little bit like, I feel a little bit that you ignore the tumor microenvironment a little bit in your thesis. So what do you think, like what could happen or which cells could influence the outcome that you find now in this single cell? How can that be, could this outcome also be influenced by the tumor microenvironment? Have you thought about it? Like, because you say in this single cell, it's look really promising, but do you think that cells within the tumor microenvironment can change this outcome or affect this outcome?
Actually, the two more microenvironment, the first thing which reached my mind is the iron level there. Actually, I think the two more microenvironment, if the iron rich or iron poor situation is there, which could really influence the treatment effect. Well, if the iron level low there, I just doubt that maybe DiAM2 inhibition can contribute nothing. For if the high iron in the tumor microenvironment, maybe DiAM2 inhibition could induce dysfunctional iron metabolism and even induce the lipid polarization or some other iron-dependent reaction. Well, I can only do my assumption.
Yes, but I think that's correct what you say, because it's, for example, know that macrophages within the TME, they also release or they take care of the iron so that you get more iron in the TME. So that's, I was thinking about like that it can be totally different when you also include the tumor microenvironment. So if you want to do that in the future, I think you can also change your cover a little bit and extend it to only one cell. So maybe that's something to do. So if I have time, I want to quickly ask you one question about proposition 8. I don't think there are online paronyms who will read it, so I will read it myself. So it's about, you say, scientific advancements requires risk-taking, inaction yields no process in this thesis. So what do you mean with risk-taking?
I want to answer the question with my research story in chapter 4. Well, when we find the increased radiosensitivity, firstly, we measure cell DNA damage. We analyze cell cycle, but unlikely there is no significant difference. But in order to understand the reason behind, we started to try ferroptosis. I think this is a risk, right? If we don't try, we get nothing. But we try, and then we definitely find, well, the increased radiosensitivity is because of ferroptosis. I think this is a risk. It's a worthwhile taking risk.
Yes, I totally agree with the proposition, and I also really like this risk-taking science. So congratulations again, and I think I give the work back to the prorector.
Yeah, thank you. Thank you, Dr. Melot. Then the opposition will be continued by Professor Zalgren. Professor Zalgren is Professor of Cell Biology at the Department of Biochemistry and Cell Biology at the University of Turku in Finland. Professor Zalgren.
Thank you very much, Mr. Prorector and dear candidate. I also want to start by congratulating you to this very, very nice thesis, and I read it with pleasure. And my main focus in my own research is notch signaling, so I will spend some time on the mechanisms of the regulation of notch signaling. So my first question is basically just from my understanding. So in all the cases where you looked at active notch, is that in a situation where you have activated notch by growing the cells on ligands?
Highly esteemed opponent, thank you very much for your nice word and question. Actually, we use a cell with the overexpression of JAG, which means the notch can be activated always. The overexpression of JAG, just we want to overexpress JAG. Why we want to overexpress this? Because we want to get a signal with the activation of notch.
So is this the same cell line, or are you talking about a core culture?
Well, the JAG we just overexpressed there, right? The JAG can be overexpressed on the membrane of the cell. when we culture the cell can attach each other, right?
Yes, I understand. So basically, this is very boosted notch signaling in a way. So have you looked in a cell line, another cell line where you have very high basal notch activity, if you see the same effect, if you would inhibit or knock down DMT1?
Actually, my previous colleagues has already finished the experiment.
Yep.
For me, actually, previously my colleague used T-cell leukemia. For T-cell leukemia, the notch tend to be there is mutation. The mutation of notch could cause T-cell leukemia. My colleague used chloroquine to block intracellular trafficking, and based on that research, we just think, well, the inhibition of notch intracellular trafficking or the damage of intracellular trafficking could influence notch activation. So
you propose that DMT1 basically affects degradation of notch or the activated cleaved notch. But if you disrupt the lysosome, so the notch can not only be degraded by the lysosome, but also activated by the lysosome. So have you looked at the impact of DMT1 on cleavage?
Cleavage or notch?
The gamma secretase mediated cleavage of the receptor.
Actually, for DMT knockout cell line, we can always get the high expression of NICD. The NICD forms the notch protein digestion by gamma secretase, right? So high NICD in the cell, which means the cleavage by gamma secretase has no issue without DMT1.
Yeah, so you override that impact from the potential lysosomal induced cleavage. Okay. Yeah. And then if I look at figure 5, and you looked at here at the different localization of the notch ICD, and you also of course have degradation that is induced by phosphorylation of notch ICD by CDK8, right? So you have CDK8 mediated phosphorylation of notch ICD that then targets it for proteasomal degradation. And I was wondering, I don't know if you look at figure B and you look at cytosol.
Yes.
It looks like you would have more notch ICD in the cytosol. Or is this just this blot? Have you quantified this?
Yes. Actually, no matter high notch protein in cytosol or not, the most important thing is we need to make sure whether notch protein can interact with binding protein RBPG.
Yes.
No matter how high NICD is there, the most important thing is if NICD cannot interact with RBPG, well, the notch, that activation complex cannot be formed. Notch cannot be activated. So
yeah, but that you have shown very clearly. So you show that the cleaved you have higher notch ICD, but you have less notch activity and you have less interaction with CSL and between notch ICD. So that I agree with you completely. So what do you think is happening?
Actually, I have many ideas which I want to do to explain these points, but because of the time limitation, why without DMT1 notch cannot interact with RBPG?
Yes.
I'm very curious about the modification of NICD. Yeah. There are many modifications which can influence this binding efficiency, like ubiquitination, methylation, phosphorylation. in addition, for RBPG, there might also be a modification. So it's better to analyze the modification of both protein in the future.
Yeah, so you think it's basically a question of deregulation of the some kind of modification.
Yes.
Do you have any experimental data? Have you done anything? What would you do to address these modifications?
Actually, we use mass spectrometry to measure the NICD modification. We can find the hydroxylation was changed without DMT1 hydroxylation. But well, we needed to double check. We needed to do Western blot to make sure the result is consistent. But unluckily, I don't have enough time to further study, so I just finalize my research.
Yeah, I understand. But you don't think it's a question of nuclear translocation. So you think that there's no difference in so the notch ICD that you detect is really, I mean, it's clear from your images that you have active notch in the nucleus. But if you have a very boosted system, you wouldn't see the notch ICD that would be cytosolic. So that's why I was so curious about the band that I saw in the cytosolic. But you think basically that the notch ICD goes to the nucleus and then it cannot bind. That's your hypothesis. So then a kind of a more general question. So this is, of course, working with notch, and in the notch field, it has been thought of as a very interesting therapeutic target for cancer. So do you think that your discoveries, but it's also very, very difficult to target because of side effects and very spatial temporal regulation. And do you think your discoveries could somehow help? Will they open a possibility to target notch, and in what way?
Well, based on my own study, if I really want to do treatment towards notch-dependent disease, like notch-dependent cancer, well, actually, I would like to try to inhibit DMT1 and then indirectly inhibit notch signal. Just provide the more possibility to achieve treatment effect. Especially well, like T-cell leukemia, if we can block DMT1 and then inhibit notch or induce dysfunctional iron metabolism, maybe we can achieve a good treatment effect.
Yeah, nice. One last thing, did you look at all at the full-length notch and where it's located in the cells, in the knockout cells?
Actually, when we want to study why without DMT notch was inhibited at the very beginning, we were very curious about maybe notch protein just blocked in some vesicles. If you analyze the full length of notch, maybe the full length notch just be blocked in early endosome or later endosome. But still the most important thing, NICD should reach nucleus. If we can find without DMT1, NICD still reach nucleus. For distribution of notch protein in early endosome or some other vesicle, we just don't care at this point. We only focus on NICD localization. So
this was for my curiosity also, so being very interested.
It's a good question.
So I think I'm up. My time is up.
It is, yeah.
Yes. So I give the floor back to the Mr. Prorector.
Yeah, thank you, Professor Zalgen. The opposition will be continued by Professor de Ruyscher. He is Professor of Radiotherapy, particular on respiratory oncology, and at the Department of Radiotherapy, Maastricht University Medical Center. Professor de Ruyscher.
Thank you very much, Mr. Prorector. And first of all, I want to congratulate the candidate, all your work, your whole team, of course, and also your family. It was already been said. It's a great investment for everybody to achieve this very nice thesis. But of course, I will ask some questions and I will focus on chapter 4. And if I look at figure 1 on page 97 and figure 6 on page 103, I have the impression that the effect of either blocking TMT1 or knockout, that you have a differential effect according to the dose per fraction if you look at the survival curves. Is this correct? Or is my interpretation wrong?
Highly esteemed opponents, thank you very much for your nice word and question. If I understand correctly, you mentioned the dose there is different, or could you rephrase your question?
Yes, with pleasure. I will rephrase my question. Actually, I have the impression that at doses of 2 gray per fraction, you hardly have an effect of blocking in one way or the other TMT1 or DMT1, whereas at higher doses per fraction, you do seem to have a small but a significant effect. Is this a correct interpretation of me?
Yes. Yeah.
And why would that be? Why wouldn't you hardly see any effect of doses of, say, 2 or 3 gray per fraction, which we typically use in locally advanced cancers? And why do you see the effect at higher doses per fraction and not at 2 gray?
Well, I think it's just because the increase of radiosensitivity is well, about that curve, we quantify the dose from 2 gray, 4 gray, and 6 gray. Finally, we needed to do quantification. From the curve, we can compare the radiosensitivity. If we only focus on the one dose treatment, I think it's we cannot compare the radiosensitivity anymore.
I see your point. You do a classical clonogenic test, and then you will have a full range of doses. But still,
Yes.
my question is, why don't you see an effect on 2 gray? And you do see a small effect, but a significant effect at higher doses. Why is that? Why don't you see an effect on also clinically relevant doses?
For high dose, the damage towards the DNA might be enhanced, right? So for cell, it's difficult to form the colony. If you give the high dose radiation, the cell cannot, the DNA damage there, and the ROS can also be generated, right? And for cell, it's difficult to survive, and then the colony cannot be formed. For 2 gray radiation, I tend to believe, that damage is not very high enough. After low dose radiation, the cell can, there is DNA damage repair, right? This can really help the cell survive after radiation and form a colony.
And why wouldn't then DMT1 don't have a function at the 2 gray, so call it low doses, low damage? Why is then that particular pathway not very important, apparently, if I understand your comments well?
Actually, we only focus on the radiosensitivity. Well, why DMT1 has no influence on 2 gray, or with treatment or without treatment? Actually, maybe just at 2 gray, the DMT1 inhibition or deletion cannot help cell, cannot damage the cell easily. Only at a high dose.
Okay, I see your point, but would you think that the notch regulation, which is very important, if you think about how DMT1 works in cells, so the regulation of the notch pathway, would that play a role, do you think, according to the dose per fraction?
Can you briefly answer this question, please?
Yes, for notch contribution to reduce sensitivity, actually, when we do rescue experiment, when we use ferroptosis inhibitor to treat the cell, we can decrease reduced sensitivity. Based on this point, we tend to believe the increased reduced sensitivity results from ferroptosis, not from notch signaling. Maybe notch signaling can contribute to increase the reduced sensitivity, but that contribution is not very significant. But ferroptosis contribute.
Thank you very much. I give the word back to Mr. Prorector.
Thank you. Thank you, Professor Ruijsen. The opposition will be continued and finalized by Professor van Zandvoort. He is Professor of Advanced Optical Microscopy at the Department of Genetics and Cell Biology, also at this university.
Dear candidate, first of all, I would like to congratulate you, of course, you, your family, your friends, and also your promotion team with being at this milestone. And as you convincingly have described in your acknowledgements, that can only be reached by very hard work and self-correction. And I think you did a great job also until now into the defense. I think you can be proud of yourself in that, and I want to say that. However, that doesn't mean I don't have questions. And the questions I have to ask you are all related, surprise, surprise, to microscopy and the types of microscopy as you describe it. One of the things I noticed is that although 50% of your images contains microscopy images, you nowhere actually, at least I couldn't find it, describe what kind of microscope you used, except that it was a confocal microscope, but nothing on the settings like filters, that kind of information. Could you extend a little bit on that? Where did you image, what type of microscope and what settings you had?
Esteemed opponent, thank you very much for your kind words and question. For the setting, we use actually I forget the number, the name of that machine. It's a Leica. Well, in our university there is 2 microscopes on the 0th floor and the 5th floor. I use both of them. For the setting, there is a web. There is a web. There, if we write down the excitation wavelength of the dye, that web can give us the setting, and based on that, we could get the the best image. For specific setting, well, you know, it's difficult to give the answer directly.
Yeah. So why I ask this is that these settings, as you describe now, are important information in the sense that it is necessary for reproduction by other people, also from your experiments. What I was referring to is the settings of the filters and objectives and the male aperture. Do you know what objective you were using?
I think objective 63. Always.
Okay, so
I tend to use that setting always, pretty often.
Because you say you did it on the SP8 cars, but as far as we know, as I know, we don't have that at the moment. So what you mean probably the SP8 stat microscope.
The machine is a 0 floor,
so
maybe, yes.
The stat microscope, okay. What you also indicate is that you image 20 cells from independent analysis. Can you indicate more details on how you did that and what type of experiments that were?
Actually, every time I capture the image for one sample, I tend to use the microscope to take the image randomly, and from one image, we can quantify the signal from the cell, and then quantify the cell from this image, from that image, and gradually we tend to quantify 20 cells.
Okay. Okay, so that was random from various images, random in the
Yes.
Okay, and the analysis you described that you do it in Imagier, but you don't describe anything on the procedure that you used. What type of analysis you did? You just say that the intensities have changed, but can you extend a little bit on the procedures that you used, the algorithms that you used for that?
Actually, for the signal, if you see it clearly, there are different types of signal. Some of them have the dots.
You can briefly answer, Mr. Zhang.
Some of them have the dots signal. Some of them only have the intensity signal. For dots, we can quantify the number of dots. For the intensity, there is the setting in the image G. We can capture the value of intensity, and finally, we can compare the difference.
Thank you.
Thank you, Professor Zandvoort. Then, Mr. Zand, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and the quality of your defense, and I request that you and your company await the result of our deliberations and our return.
Thanks. See you later.
see. So, Mr. Zhang, can you hear me? You can? Mr. Zhang, the degree committee here present, the degree committee here present has assessed the quality of your thesis and the quality of your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Voice is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor. Professor Voice.
Thank you, Mr. Prorector. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Rui Zhang, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university, shown by the beadle. You can see that now. And we will soon sign this, and this will come your way. I give the word back to the prorector now, briefly.
Yeah, but you can continue with the laudation.
So I have the honor to read the Laudatio. Dear Dr. Zhang, dear Rui, I'm pleased to be the first to congratulate you on obtaining your PhD and addressing you as doctor, but of course also on behalf of your co-promoter Tom Curlers. We first met through the Chinese scholarship program. I interviewed several candidates, and your profile stood out for your curiosity, enthusiasm, and motivation. We developed a 4-year project together, submitted the application, and waited. And after several months without an answer, we assumed it had not been selected. Later, I discovered that due to an administrative error at our university, the proposal had not been considered. And when the next opportunity arose, I contacted you directly, and we submitted again our proposal, and this time we succeeded. Your PhD started in October 2021. Your main task was to identify the mechanistic basis for reduced notch function in the absence of the iron transporter DMT1. Many techniques were new for you, and setting up the assays took time and troubleshooting. There were setbacks, including issues with contaminated cell lines, and some experiments had to be repeated from the beginning. And as in most research, not all initial observations could be reproduced. Despite these setbacks, you stayed focused and kept moving the project forward. With support from Yolanda and your colleague Somaya, whom both made important contributions to your findings, you developed a clear story showing how disrupted iron homeostasis affects notch transcriptional regulation and protein turnover. With your perseverance, you overcame problems and obtained reproducible, quantifiable, and publishable results. And I'm happy to add that last week we heard that your paper was accepted for publication in Fab's journal. During the first 2 years, you were also supervised by Florian Bock, who later moved on to another position. But Florian and you worked closely together in the early phase, and he deserves credit for his supervision during those initial years. As the project moved into lysosomal biology, an area that was relatively new for our group, it was helpful to have Tom Curlers as second supervisor and an expert in this field join the team. Together, you wrote a nice review on lysosomal function, which supported the interpretation of our data and helped us to refine and develop new hypotheses. And finally, you extended the work towards a translational question, whether DMT1 loss through altered iron and Rod signaling could increase radiation sensitivity in vitro, potentially involving ferroptosis. That work is not yet published and still requires follow-up experiments, but it forms a relevant chapter in the thesis, and you were also supported by Maika and Chuchi and Yolanda in this work. During your PhD, you had many opportunities to present your work to local, national, and international colleagues. 2 meetings stand out: the Wolfsburg meeting for molecular radiation biology in Norway and the International NOTCH meeting, the Gordon Research Conference in Maine, USA. You also had the opportunity to give a presentation to your peers at the pre-meeting and had the full dorm experience at Bates College in Lewiston, a top-notch meeting in which I've been to many times myself, but unfortunately could not join you at that time. Life is a journey is Rui's motto. You can even find it on his Instagram page. And the cover of his thesis also suggests a journey around the world, at least halfway, and galaxies, but also a journey around the cell and perhaps inside the cell. The rocket can symbolize several things: rocket science, but also being launched into a new environment, from one culture into another. And probably it means all of the above. It was not always an easy journey, but those are often the ones you learn the most from and remember the longest. Along the scientific journey, you made also the most of your time by traveling wildly across Europe, the UK, Austria, Portugal, Spain, Norway, and Italy, and even in the United States, including Boston and New York City. So today we celebrate Rui, not only for completing his PhD, but also successfully surviving Europe. Because honestly, the past few years have been a research project and training, but also a long-term experiment in adaptation, resilience, and creative problem solving. So let me take you through a few chapters of Rui's survival guide to Europe that also has some interesting chapters. First chapter, nutrition. One of Rui's earliest discoveries in the Netherlands happened in the supermarket. Most people compare labels, check ingredients, but Rui just grabbed a carton that looked like milk, went home and drank it for breakfast. Only later did we realize he had been starting his day with full whipping cream. He did mention it felt very filling. Well, yes, Rui, that's because it was 35% fat. But hey, he survived. Stronger, actually. Chapter 2, housing logistics. When Rui had to move houses, he took a very academic, very analytical approach. He moved his entire household into one of the offices for a few weeks. A mini washing machine, check. Furniture, check. Half his worldly possessions, also check. Most people would call that chaos. We call it field data. And to be fair, Rui carried everything himself. No help needed, pure determination, and maybe a little confusion about European moving services that most of us use. Chapter 3, mobility and transportation. At some point, Rui injured his leg, maybe even broke it, and had to use crutches. I was really worried for a while, not sure whether he was taking sufficient rest and correct advice from the doctors. And most people, that would be the end for cycling for a while, but not for Rui. He simply continued with crutches, did his grocery shopping, and carried on as if Newton laws did not apply to him. And this is the moment we realized Rui wasn't just surviving Europe, he was outsmarting it. Chapter 4, social adaptation. Of course, survival is not only physical, it's social as well, and Rui mastered that part too. Whenever we were out, he loved dancing. Not the shy 2 steps to the left, 1 forward kind of dancing, but full energy, joyful, I'm having the best time of my life. That energy was contagious and also a great indicator that he had successfully integrated into the European nightlife culture. Chapter 5, culinary choices, but perhaps the most fascinating chapter of his survival guide, how Rui prepares food. We discover that Rui heats up supermarket pizzas, the ready one makes, in the microwave, not the oven, not even by accident. This was a deliberate strategy. To this day, we are still analyzing this phenomena, but at least he has taken also the like of the tosties, the RT Lab menu special. Finally, chapter, who Rui really is. All jokes aside, Rui is genuinely one of the kindest people you can meet. Curious, hardworking, helpful, and always trying to do his best, whether in the lab, navigating Dutch culture, or traveling across Europe. He loved exploring new places, learning new things, and even brought his own cultural contributions, like homemade limoncello and his occasional bottle of cry wine at the office. Rui, you didn't survive Europe, just survive Europe. You embraced it, you challenged it, you confused it, and ultimately you conquered it. Congratulations on your PhD. You worked incredibly hard, and we are all very proud of you. You are still in the process of finalizing your visa for your postdoc in Boston in the USA, where you have been accepted for a position, and we hope this is soon resolved for you. So for now, as you enter the next chapter of your life, please remember, in Europe, pizza goes into the oven. It's really better that way. We wish you the very best of luck, and drop by if your rocket needs refueling. Take care.
Thanks very much for your help. I will go back to say goodbye. Well, see you again.
No. Professor Dr. Zhang, highly esteemed Dr. Zhang. It is my honor and pleasure to congratulate you on behalf of the University of Maastricht with the degree that you have just acquired. I also would like, of course, to congratulate your supervisory team, Professor Boyce and Dr. Keulers. I would like to congratulate your family, your friends, and also those that have collaborated with you on the projects that were described, have been described in your thesis and will be described in the publications that come from your thesis. I think you have done a good job. I can say a little bit about our deliberation. All the members of the opposition committee were very pleased by your very nice and clear presentation in the beginning, very nicely illustrated also. The thesis describes a complicated topic, and I think it was brave of you to embark on this project. And I think despite the different, the lot of questions that remain, this is a topic that will be very important for future treatment of cancer. With respect to your defense, all of the opponents thought that this defense was satisfactory. It took you some time to get into the mood, and but during, let's say, during the discussions, your answers became better. All over, I obtained the feeling that you are a very enthusiastic researcher, and therefore I also would like to wish you a lot of luck in your future research. As you state in your CV, you would like to continue your academic career in the biomedical field, and I hope that you will succeed so in the Boston area, where apparently there is a postdoc job for you. So lots of success for the future career, for your future career, and again, congratulations. What I would like to do now is to give the opponents a chance to congratulate you, and I will start with Professor De Wind.
I can be very short, Dr. Zhang. So congratulations, you did fantastic. I knew I caught you a little bit off guard with the cover of your thesis, and but whenever, maybe in this long flight going to Boston, in the long plane flight, you can buy the book, The Little Prince. Remember that, The Little Prince from Saint-Exupéry. It's really very touchy. It's very emotional. It's an emotional book. But when you see the cover, then you understand what I mean with the similarity with your thesis. I really enjoyed listening to you. And I wish you all the best. Please be careful in the United States. Yes? Be careful.
Thank you. Thank you.
And Professor Van Kempen?
Dr. Zhang, congratulations with your promotion. I really enjoyed reading your thesis. It has so many angles for discussion, and I regret I only had 7.5 minutes. I have the feeling that afterwards we could go on and on to discuss different aspects. Again, I applaud you on taking on this difficult subject, and I wish you the best of luck in Boston. Please be well.
Thank you. Dr. Milot?
Yeah, also congratulations, Dr. Zhang. I was also very happy to hear from Professor Boyce that next to science and hard working, that you also really enjoyed the good things in Europe, and that you travel a lot and that you have the opportunity to do other stuff than only science. And I would also suggest you keep doing that and enjoy your time in Boston, also next to the science. So good luck.
Thank you. Professor Zalgan?
Dr. Zhang, my warmest congratulations to you. It was very interesting to talk to you and discuss today. And I was wishing all the time that this was a finished defense where we can go on for 5 hours. But unfortunately, we only had 7.5 minutes. But it raised a lot of questions and very, very interesting thoughts. And I wish you all the best in Boston, and I hope you use your skills to survive also over there.
Thank you.
Thanks.
Professor Deliberation?
I was also very pleased to read your thesis. I learned a lot by reading your thesis, by the way. And indeed, I would like to have a longer discussion with you, but I'm sure that you will proceed with research in Boston at a very high level. So a lot of success there.
Thank you. Professor van Zandvoort?
Well, thank you again for your presentation. It was very convincing, and congratulations, of course. And also from the Laudatio, I had the impression that we should have learned to know you better because it was very interesting, I think. And you also make a very positive and strong impression. And I wish you all the good luck in the States, and I'm sure with your personality you will succeed.
Thanks.
Thank you. And then finally, since Professor Voice has already expressed his laudatio, I would like to give the word to Dr. Keulers, your supervisor.
Yeah, Rui, congratulations on your successful defense today. Of course, completing a doctoral project is long and was a demanding journey, but I think you carried through with a lot of persistence and curiosity and your genuine passion for your topic. So your defense today not only showed your expertise, but also I think your growth as an independent researcher. So you can, I hope you can look back on this with pride, and it marks the beginning of a new chapter. So all the best to that.
Thank you all. Professor Voice, would you like to say a final word after your extensive laudatio?
Well, I think it was a truly joyful and learning experience for both of us, also for me. So I thank you for that as well. And yeah, of course, after everything that has been said, I can only now wish you the best of luck in the US. And please stay in touch as a researcher, but also as a person. I really enjoyed working with you, with the team together on this interesting topic, which will continue in my lab. And well, thank you again. And of course, congratulations with what you have achieved today, your doctorate, but that speaks for itself. So best of luck.
Thank you.
And with these words, I end this ceremony, and thank you all for being present, and say goodbye.
Bye-bye.
Bye.